Hello students, let us now come to the lecture 5, which is basically the continuation of the previous lesson. So, in the previous lesson what we have discussed about the production of specialized metabolites by plant cell and tissue culture, and I have ended with one of the important strategies, that is elicitation, which is used to uplift or produce enhanced level of metabolites. So, this lecture 5 is the eliciting specialized metabolism in culture. So, that is our main point of discussion. Let us now go to the slide. The concepts what will be covered here is the rationale for elicited treatment, why we go for elicitation, and then elicitation of in vitro grown cultures, so that means cell cultures or shoot cultures. Then we will talk about the mechanism of elicitation. Then also we will talk about the elicitation of ex vitro plant organs. That is that you can also, it is possible that you can elicit a plant organ not inside the culture, but even outside the culture as well. And then we will also talk about a model for jasmonate induced upliftment of alkaloids. So, jasmonate here act as an elicitor, and how that upliftment occur and at the molecular level, we will also I will also discuss a little bit about that. So, in the last class we talked about the production of cell and organ culture, particularly cell culture, root culture, shoot culture. So, one important point is that why plant cell culture is so attractive, because of its visual appeal. Nobody can deny that. You see, this is a lump of callus, this is a grown suspension culture, this is another thickly grown suspension culture in bioreactor, this is in the flask. So, the with this visual appeal, scientists started thinking or predicting that lot of things can be possible to make if we can exploit the cell cultures. That means, what they thought, cell cultures is really something has been achieved. So, these cell cultures we can manipulate and we can make metabolites whatever these plants are producing. So, there are a lot of attraction, lot of positive air, that time it was blowing. So, that a lot of investments were made to make cell cultures as a mean for producing metabolites, but of course, over the years what has been found, that cell cultures are not that attractive, because what important point I mentioned, that there is a problem of instability. That is one point. And second, another important point to tell you here, that a metabolite normally if it is produced in the root or in the shoot, in the cell cultures as the life cycle is totally different, it is totally compressed or not a proper life cycle is maintained. So, the metabolites which are synthesized in the leaf or root, okay, cell cultures may not be the appropriate means for synthesizing these metabolites. That means, in other words, there is a need for differentiation. There is a need for differentiation of leaf or root tissues. When you talk about the differentiation, there is a lot of changes in the structure of the tissue. Different type of tissues are coming here, very specialized cells are produced there. For example, I mentioned in one of the previous classes, like latex producing cells, like oil cells. So, these are very important, because these metabolites sometimes they are toxic. So, it has to form only inside the cell. So, cell culture some success are there, but often they fail to produce these specialized cellular structures which are found in the plants. So, then how can you go ahead with that? But even then, cell culture, the good point is this, that you can control their growth, you can grow faster, you can grow all around the year, and it is possible to manipulate the cell cultures by empirically like changing the medium composition by adding certain components into it, okay, by challenging with elicitors, which I have mentioned just in the previous class. So, in other words, that if cell cultures can be elicited by foreign molecules, then what happen? These cell cultures may work like the cells which are produced in the normal fully grown plant. That means, this cell culture, if these cell cultures can be challenged with foreign molecule, these cell cultures will evoke a defense response. In other words, specialized metabolic pathways will be activated. So, that was the rationale for choosing elicitation. Now, come to this example Catharanthus roseus. So, very important plant, all of you know, or this is sometimes called vinca rosea, it produces these vinca alkaloids. So, vincristine and vinblastine, these are two anti-cancer drugs used for treating the leukemia patients. And they are produced by two industries, and this is the structure is so complex that chemically synthesis is not at all feasible at commercial scale. So, these metabolites are harvested from the aerial part or leaf tissues. Okay. In tons and tons of leaf tissues are processed, and then only you will get a few milligrams of this highly valuable anti-cancer drugs. That is why they are so expensive. So, the question came to the mind of scientists that if cell cultures can be developed from this leaf tissue, and first question is that whether cell cultures are capable of producing that. If not, then can we challenge the cell cultures with certain elicitors, so that the metabolism can be uplifted and we will be able to produce these metabolites at a reasonable scale. So, these are the questions, and these are the questions behind the drive for moving into the elicitation work. So, elicitors are foreign molecules which when you add in the cell cultures. So, the cells basically, as I said, treat them as foreign molecules, and in the cell membrane there are certain receptors. Receptors are nothing but a type of proteins. So, which will basically recognize this elicitor, and once they recognize this elicitor, then there will be certain metabolism activated. So, that we will talk bit later, and as a result of that there will be defense response, induction of defense response. So, this defense response, there is lot of physiological changes happens, including the expression of genes related to the formation of these defense compounds, which are the specialized metabolites, and which again are the bioactive compounds which we are looking for. So, this is basically in a nutshell the impact of elicitation on specialized metabolism. Now, here I will show you the list of elicitors used, abiotic and biotic. So, abiotic like chemical, as I said, heavy metals, even nano oxide, acetic acid, physical like UV, ozone, PEG, light intensity, hormonal, salicylic acid, jasmonate, methyl jasmonate, nitric oxides. So, these are well proven elicitors, and also the biotic elicitors includes the mycelial extracts of several fungi listed here. Also, the saccharides like chitosan, chitin, pectin, these can also be used. In addition, the cell cultures can be co-cultured with some of the bacterium species, and that sometimes resulted in the evoking defense responses, and you may get the desired metabolites at the uplifted level. So, this is the Hyporhizum perforatum cell culture. So, Hyporhizum is producing hyperforin and hypericin. So, Hyporhizum is a very well known herbal drug available, and particularly western world Europeans are very fond after Hyporhizum. So, and we will talk about Hyporhizum later when we will come to that particular group, but here what I meant to say here, the first the Hyporhizum perforatum cell cultures were established. So, what is showing here, and later these cell cultures treated with elicitor, and as a result of elicitor what happens, the cell culture started changing its color. So, it is producing red color. So, this red color could be because of the metabolite hyperacin and other compounds. And if you see the phytochemical profile here HPLC, so when it is in the unelicited condition, that is this bright green yellowish color. So, you see only here 1 or 2 peaks, and once you treat it with elicited, then you will see multiple peaks. So, these multiple peaks means that it produces new metabolites which are not produced in unelicited cultures, and these can be collected, fractionated and can be used for testing, for bioactivity screening and leading to drug discovery. So, it is possible to separate these pigments at preparative level by using preparative HPLC, and individual fractions can be extracted and identified and can be tested for biological activity. So, the molecular mechanism of elicitation I can now tell briefly, that when elicited molecules are basically recognized by the receptor here. So, that once it recognized, it basically leading to certain changes in the plasma membrane bound receptors, including there is a rapid rise of calcium burst, then there will be ROS burst as well as activation of G proteins. Not only that, so when there is a ROS burst, then it produces the H2O2. So, this H2O2 of course will play an important role, but what is that is not only in ROS, but this H2O2 can also activate the other pathways leading to the higher expression of the genes. So, in other words, both ROS burst, G protein activation, H2O2, they activate the MAP kinase enzymes, that leads to activation of certain signaling molecules, including jasmonates, and as a result of that, it activates the several transcription factors, and that again activates the specialized metabolite genes, and eventually it produces the enzyme proteins in higher amounts. This is very important, enzyme proteins in higher amounts, and then as a result of that we will see the response of specialized metabolism at uplifted scale. So, this is basically in brief the mechanism of elicitation. Now, I will talk more about the practical aspects. Okay. So, this is the biosynthesis of terpene indole alkaloids in Catharanthus. What I said, that this Catharanthus roseus produces dimeric indole alkaloids like vincristine and vinblastine, which are anti-cancer drugs. Apart from that, it also produces azmalicin, azmalin, serpentine, which are also used for treating different ailments, including your control of blood pressure and blood related diseases. So, the pathway is very complex, which of course, we will study when we will study details about the metabolic pathway for indole alkaloid and metabolic engineering, but so it originates one from the secumate phenylpropanoid and the other comes from the terpene skeleton, and these two, one that is the secologanin is coming from the terpene moiety and tryptamine is coming from the secumate pathway, and these two basically joins and form strictocidin. Strictocidin is the first step of this indole alkaloid synthesis, and then strictocidin will be there will be a glucosidase activity, which the glucoside will attach, and then again there will be breakdown of glucose, that will be a glycone, and then there are multiple reactions anyway. So, that was so the cell cultures of Catharanthus roseus was established long back, and in fact, there are plenty of studies you can find with Catharanthus roseus alkaloids. Now, more than 100 publications, even more, are available with indole alkaloid production and the problems, hurdles. Okay. So, and this is picture is showing that cartoon picture, that you are adding elicitor and that elicitor may enhance the metabolite production. Now, what I said here, that in Catharanthus roseus, that when they try to produce the indole alkaloids in higher amounts, one of the elicitors they have used is jasmonate or methyl jasmonate. So, when jasmonate is added in the cell culture, that will be basically recognized by the jasmonate receptor which is there in the cell membrane, and once it is recognized, then along with the activation of this G proteins, ROS and MAP kinase, eventually it activates transcription factors specific to jasmonate. So, what happens that in the DNA of the cell cultures there are jasmonate responsive elements were there. There is a transcription factors which getting activated, and once it is activated, that in turn will activate another transcription factor which is called ORCA3, that is specific for the indole alkaloid production, octadecanoid responsive catharanthus AP2 domain. So, ORCA2 once it is getting activated, it bind in the jasmonate elicitor response element, and that in turn activate the strictosydin gene expression. So, strictosydin gene expression leading to the formation of strictosydin. So, strictosydin synthase, this STR is first getting activated, uplifted upon jasmonate treatment, and then it produces strictosydin and subsequently several indole alkaloids. So, this mechanism has been fully elucidated at the molecular level. We will again come back to this much later when we will talk about the alkaloid pathway and metabolic engineering. Now, apart from this, there are other very interesting examples with the root cultures. So, here what I am showing here, there is another one, the Canskora diffusa, Canskora decaceta, both the species which is producing these xanthone metabolites. So, we have the root cultures of both. So, these root cultures are established, and once the root cultures are established, both the species we elicited with here we elicite with it chitosan or sometimes with the yeast extract. So, the autoclaved yeast extract, and as a result of elicitation. So, this is basically the control profile. So, these are HPLC peaks which including the compounds, and if you see this peak here and this peak here, you will see there is the height is much more. This peak here and this peak here, height is much more. That means that the compound produced in the unelicited root culture and in the elicited root cultures, there is a difference, and elicited root cultures are producing more metabolites, and when we measure that in terms of total xanthone content in the root culture, this is a different one. That means that normal the xanthone metabolites are produced more in the root than in the shoot. So, basically it is showing that, and that is why root cultures were established. Root cultures, the rationale for establishing root cultures, because it produces more xanthone, and these root cultures were elicited, and what we found that root cultures producing more xanthones. Another good example was Hopia. So, Hopia shoot cultures were treated with yeast extract, autoclaved yeast extract, and as a result of that, what happens that there will be a rapid upliftment of the xanthones. So, the bottom one is basically the control one, and this is the upliftment, and the red arrow indicates that how much rise in the metabolites. So, these particular xanthone metabolites, which basically are important molecules to treat nervous ailments, particularly they are the good inhibitors for acetylcholinesterase and monoamine oxidase A and B, which overproduction of which leads to the Alzheimer's and Parkinson diseases. So, this of course, these elicited metabolites were isolated from semi-preparative run of HPLC, and individual metabolites were tested in vitro for this inhibitory activity, and it showed really very good promise. And later the mechanism of elicitation has also been elucidated for the Hopia, and where it was shown that H2O2 plays an important role in uplifting the early steps of the biosynthesis leading to activation of the genes of phenylpropanoid and xanthone pathway, and that leading to more formation of xanthones. And when more xanthones are produced, what was also found that it limits the phenylpropanoid pathway flow. That means, there what maybe at some later stages we will see that the PAL which moves the pathway in this direction, it got suppressed, instead this pathway got activated. That was indeed very interesting. So, lot of basic information can also be generated from the elicitation studies, although the focus will be on the practical aspects, but when you try to understand the science behind it, it is possible to generate a wealth of information on the basic sciences. Now, this is now the another part. So, here there is a plant called Ananthmul or Hemidesmus indicus. So, the plant is fragrant, not the root is fragrant, not the upper portion, and the dry roots are available in the herbal shop for multipurpose uses. So, it is a well known source for purifying the blood, but the roots are fragrant, and particularly in the southern India and Tamil Nadu districts, part of Kerala, the dry roots are sold as such, and as well as a processed product called nannari. So, dry roots are extracted and they add sugar syrup and they make a flavored drinks which is called nannari, and which basically cool down the body. So, that the important point is that roots are fragrant and the fragrance are pretty similar to that of vanilla. So, that of vanilla, that means the vanilla has a typical fragrance, and this plant also has a fragrance which is pretty similar to vanilla, although the compound is slightly different. Here we are not discussing that aspect. So, the point here is this, that once it is producing the fragrance, actually it raised interest to the scientist. So, can we really make this fragrance in higher amount in roots? So, the problem is this, that the proper convincing root cultures were not available. So, what was available is basically you dig out the root from the field and then you wash the root, cut that into pieces, and then first you check for the fragrance compound which is called M-balt as a methoxybenzaldehyde, and once you detected that, then what was attempted, that can we really make more fragrance in this excise root. So, that was it, that is called the root ex vitro. So, that was possible by treating with elicitor. So, in other words, these excise roots were treated or were elicited with chitosan or yeast extract or methyl jasmonate, and as a result of that, here the picture shows the whole plant, but ideally it should be only the excise root, and as a result of that, there is a time course there was a time course increase of this M-balt, which is the principal fragrance compound accumulating in the roots. So, that means this ex vitro elicitation of Hemidesmus indicus root, excise root was very successful. So, the approach was that excise field roots were used for elicitation, and then they were treated with either chitosan of yeast extract, and it was incubated for 12 to 24 hours, and after incubation the metabolites were analyzed by HPLC, and then it was analyzed compared with the control and see if there is enhancement or there or not. So, in the previous slides I mentioned about the mechanism of elicitation. So, we are not going into that, rather let us see the result. So, a range of different elicitors were treated, for example, yeast extract, chitosan, methyl jasmonate and control. So, as compared to control, you see every elicitor treatment that showed upliftment of the metabolite or enhancement of the metabolite. So, maximum result obtained with the yeast extract, that is why the autoclaved yeast extract was used for the subsequent experiments. And what has been found, that when you start the experiment at the 0 hour, that is without elicitation, then after 12 hours and then after 18 hours. And what you see, that this is the principal peak of the metabolite So, here you see the at 0 hour this is the basal level of M belt, and 12 hours there is an upliftment of M belt, and 18 hours there is a further upliftment. That means, with time of elicitor treatment there is an upliftment of activity, and why this is activity, if you look into the science, that is only possible if the enzyme activity is more. So, when the enzyme activity was tracked along with the M belt production. So, there was a rapid burst of enhanced activity just before the highest M belt accumulation. So, first there will be enhanced enzyme activity and there will be the product. So, upon elicitation what is happening, that the amount of enzyme protein will be enhanced, because substrate supply will be limited, and as a result of enhanced protein. So, whatever substrate available, that protein will be try to utilize it maximum, and that leads to the formation of the product. So, this will be clear here. So, what has been shown here, that this is a time course accumulation of M belt maximum at 18 hours and then it getting declined. Along with that, when the total protein content was checked, the protein content also getting enhanced. That means, that enhanced total protein content means that it includes the enzyme proteins also, which is responsible for pushing the pathway. And this is one example of one of the early enzymes of the cyclimate pathway, which is called cyclimate dehydrogenase, and this is the in gel assay. So, this basically showing the amount of the active proteins detected in the gel. So, what you see here, that when you treated with the yeast extract compared to control, this is a maximum protein is produced, and this is by different treatments, inhibitor treatment. So, the pathway is blocked. So, accordingly the enzyme protein is blocked. So, from there it is clear that protein contents are enhanced in tissue culture upon elicited treatment, and that leads to upliftment of metabolite. So, the future trend will be towards increasing the screening of elicitors, more elicitors and their empirical use, inducing the synthesis of valuable metabolites. And there are several important questions to be answered, that how does an elicitor affect a specific phytochemical response. So, are specific second messenger molecules involved in the elicitation of a particular molecule, that needs to be elucidated, that is again a fundamental science. And then full molecular mechanism may need to be addressed in details. Of course, that will take years to elucidate. So, with this it is possible to know the detailed science of elicitation, and of course, the empirical approach will be continued, because elicitation treatment enhance the metabolite, and the metabolite upliftment and pathway can be studied, but exactly what happens in the elicitation mechanism. So, that also needs to be elucidated. So far, of course, few information are available which I have shown, but still there are a lot of things which we need to learn. With this, thank you very much.